the host-parasite interaction. Okay, so here is the outline of our discussion. Okay, so let's start off with definition of terms. So symbiosis is defined as the association of two organisms that are living together. So the organisms in this scenario are called symbiotes. Now, symbiosis is simply a general term that refers to a close and prolonged association between two organisms of different species. Okay? So when we say symbiosis, hindi pa po natin ini-specify dito whether the organisms are benefiting or causing harm and all that other stuff. But when we say mutualism, ito na po yung merong clear definition ng kung anong napapala ng bawat organism in the association. So ayun po. In mutualism, both the host and the bacteria benefits from one another. So kapag sinabi mo sa seatmate mo na, pera mo ako ng highlighter, tapos pinahiram ka niya pero sinabi niya, sige, pero pera mo ako ng extra mong ballpen, that is kind of like mutualism because you both got something useful from each other. Okay? So an example of mutualism in the human body is the presence of lactobacilli in the urogenital tract of women. So the lactobacilli would provide the host protection by preventing colonization of pathogenic species at the same time while they are deriving nutrients from the host. So naprotektahan nila yung host at the same time nakakuha sila ng nutrients from the host. So that is an example of a mutualism. Next, in commensalism, the microorganism benefits from the host, but it does not bring any beneficial nor harmful effect to the said host. Okay? So kapag sinabi mo sa seatmate mo, pera mo kong ballpen, tapos pinahiram ka niya, pero wala siyang hiningi in return, and hindi mo naman siya naiistorbo, that is kind of like an example of commensalism. Okay? So, in the gastrointestinal tract, ang example po ng commensalism is Proteus mirabilis. So, this is considered as a commensal because it is living in our GIT rent-free. Okay? But it isn't causing any disease as long as it stays within the GIT. Okay? So, the photo on the left is a culture of Proteus mirabilis showing its characteristic swarming motility. So, isa po yan sa mga commensals ng gastrointestinal tract of humans. Another term that we need to be familiar with is colonization. So, dito muna tayo sa commensalism. Pag sinabi pong colonization, this simply means that the growth of microbiota on or in a specific body site without causing damage or producing any symptoms. Okay? So again, colonization, this is the growth of microbiota in or on a specific body site without causing damage, without producing symptoms. So it's pretty much what a commensal organism usually does. Okay, third is parasitism. Okay, so parasitism, this is probably something we're all already familiar with. Parasitism is when an organism benefits from the host and it harms them at the same time. Okay, now microorganisms that are commonly found in or on body sites of healthy persons are called normal microbiota or indigenous microbiota. Okay? So the different body sites may have the same or different microbiota depending on different conditions. Okay? So some microorganisms that colonize an area for months or years represent the resident microbiota or resident flora. Okay po. Whereas microorganisms that are present at the site temporarily represent transient microbiota. So as the name implies, hindi po nagtatagal yung mga transient flora. They are either eliminated by the host's immune defenses or by competition with the resident normal flora. Now, a change in environmental conditions can predispose an individual to infection by those indigenous biota. So, a type of infection that is referred to as opportunistic infection. Okay, so kapag po yung normal flora nyo, because of a change in their usual growth environment, pwede silang mag-overgrow. And yun po yung nagiging sanhi ng mga opportunistic infections. So, pwede pong dahil immunocompromised yung pasyente or dahil kumonti yung population ng iba pang normal flora ng pasyente, leading to the overgrowth of the opportunistic bacteria. Or pwede rin naman po that they have been moved from their original site that they usually inhabit. So there are numerous factors as to how a normal flora could eventually cause an opportunistic infection. Okay, now some pathogenic organisms may establish themselves in a host without manifesting symptoms. Naintindihan po ba? So, pathogenic talaga sila. Pero, they were able to invade the host without causing any symptoms. Okay? So, now, these hosts would be called carriers. And these carriers are still capable of transmitting the infection. So, the condition of these hosts is called the carrier state. So, the carrier state may be acute or short-lived or transient, or the carrier state may also be chronic, which could last for months, years, or even permanently. Okay, so an example of chronic carrier state is found in post-Salmonella typhi infections. Okay po. So, Salmonella typhi, this organism can establish itself in the bile duct and can be excreted in the stool in over years. Okay? So kahit gumaling na po yung pasyente, even if there are no more symptoms from the infection that has passed, the organism may still be continuously shed in the patient's stool. Kasi po, nagtatago sila dun sa bile ducts ng pasyente. Here we have two examples of organisms that could cause an acute carrier state. So, sandali lang po, hindi sila nagtatagal, hindi umaabot ng months or years yung colonization nila dun sa host. First is Neisseria meningitidis. So, this can be found on the nasopharynx of asymptomatic individuals during an outbreak of meningitis. But after a few days or weeks at most, these individuals may no longer harbor the organism. So, kapag nagka-outbreak po ng Neisseria meningitidis sa isang lugar, most likely naman po yung mga carriers, after some time, hindi rin po nagtatagal yung organism. Another example would be Staphylococcus aureus. So, it can colonize the person's anterior nares and this is transmitted by the hands. So, when you touch your hands, some organisms may transfer in your hands, but you're only carrying the organism only after until you wash your hands. Okay, so ang gulo ng explanation na yun. Bale po, parang naging carrier kayo ng organism, but after you wash your hands, wala na po sila or na-eliminate nila sila. So that is that's also an example of a carrier state. Okay, now, which microorganisms are present at a particular body site is influenced by nutritional and environmental factors, such as the amount and types of nutrients that are available at that site. Okay po? So kaya nandyan yung mga normal flora natin at these specific sites where they currently are is because these are the sites that meet their preferred growth conditions. So first example we have here is Propionibacterium acnes, which is now currently known as Cutibacterium acnes. So pinalitan nila yung genus name. Anyway, Propionibacterium species, kasi po, although lipids and fatty acids are bactericidal to most bacteria, ulitin ko po yung statement na yun, lipids and fatty acids are bactericidal to most bacteria. Pero po, for Propionibacterium species, they are able to colonize the ducts of hair follicles because these bacteria are able to break down the skin lipids or fatty acids. So para po sa mga Propionibacterium, sa mga hair follicles yung optimal place for their growth. Kahit na for some bacteria, yung high fatty acid content ng area na yon may be harmful. Okay? So kaya po nako-colonize nung Propionibacterium yung hair follicles natin. Another example is Bifidobacterium. So for Bifidobacterium, human milk has a high lactose concentration and is usually about at a pH of 5 to 5.5. Okay? So this is an environment that is supportive of Bifidobacterium. Kaya po, yung mga bata na breastfed, it is expected that they have a greater number of Bifidobacterium in their fecal flora compared to children who were mostly fed with formula milk or cow's milk. Okay? So yun po, nagkakaroon ng increased colonization of Bifidobacterium sa mga fecal samples ng mga bata who are breastfed because the human milk was able to produce a optimal growth condition for Bifidobacterium. Now, last one, as for Bacteroides and Fuso, gusto po kasi nila yung mga places that are only capable of supporting organisms that are capable of fermentation. So, usually, they prefer sites that are the best for anaerobic organisms, okay? Just like our gingival crevices. So, the gingival crevices of humans are typically colonized with Bacteroides and Fusobacterium. So, normal po na sa oral cavity ng mga healthy individuals or healthy humans, may ma-isolate tayong Bacteroides or Fusobacterium species. Okay, let's now quickly go through the composition of normal flora at different body sites. So, tignan lang po natin kung sino-sino ba yung mga bacteria that may be commonly recovered from the body sites that will be enumerated. Okay po. So, normal skin has numerous mechanisms to prevent infection and also to protect the underlying tissue from invasion by potential pathogens. So, bale, parang first layer of protection natin against pathogenic organisms, yung skin natin. Okay? So, these mechanisms include the physical separation of microorganisms from the tissues. Also, kasama na rin po dito yung production of fatty acids that inhibit many microorganisms. Nabanggit ko po ito kanina, di ba? Nung na-mention natin yung colonization of Propionibacterium acnes. Now, ayan, kasama din dito yung excretion of lysozyme by sweat glands. So, yung pawis po natin, nagko-contain sila ng lysozyme. And there is also the desquamation of the epithelium. Desquamation, ito po yung shedding of dead skin cells. So, kapag meron man pong pathogenic organism na nasa ibabaw ng skin natin, as we desquamate our epithelium, matatanggal din po sila or sasama sila doon. So, ayun, consider din siya as a mechanism to prevent infection. Okay? So, kahit medyo marami po tayong normal flora on the skin, they don't usually bother us thanks to all the above-mentioned mechanisms. Okay, so now the normal flora of the skin depends on the activity of the sebaceous or sweat glands. So organisms concentrate the most in areas that are moist, such as the armpit, the groin or perineum. So sweat glands in those areas secrete substances that can be metabolized by bacteria. And after the metabolism of bacteria by those substances na na-release ng skin natin, this could create odorous amines. So, yun po yung nagiging cause ng body odor. So, may bacteria sa isang site such as the armpits. Tapos, the armpits or the skin on the armpits would release substances na kayang i-metabolize ng bacteria. So, after they metabolize that, this could release odorous amines. So, yun po yung dun nanggagaling yung smell. Okay? So yun po yung konsepto or yun yung how body odor is produced. And for these ones, sinama ko na lang po yung mga tables na makikita sa reference textbook natin. So, i-familiarize nyo po yung sarili dun sa mga commonly isolated organisms or commonly found organisms on the skin. Baka po mabigyan kayo ng multiple choice na ini-enumerate lahat ng to, tapos kailangan yung ma-differentiate kung sino yung hindi kasama, yung mga ganun po. Next, the mouth contains large numbers of bacteria, with Streptococcus species being the predominant genus. Okay? So sa oral cavity po of normal healthy humans, mostly Streptococci po yung normal flora natin dyan. Now, many organisms can bind to the buccal mucosa and tooth surface. So, itong mga bacteria po na nakalista dito sa box 2-2, ayan, these are also the ones that are responsible for the build-up of plaque on the teeth whenever a patient skips brushing their teeth. So, minsan po, kaya mahalaga yung oral hygiene or just hygiene in general. Kasi po yung mga normal flora din natin, yung nagdudulot ng mga bagay tulad ng tooth decay, plaque formation, body odor, yung mga yun po. So although they could help us in preventing the colonization of pathogenic organisms, kapag pinayaan din natin sila na mag-overgrow, may mga consequences din po yun. Okay, next po for the respiratory tract, the mouth, the nasopharynx, and the oropharynx are colonized predominantly by virulent streptococci. So, sa next week po yata, magiging topic po natin lahat ng Streptococcus species. Now, virulent streptococci, this includes Streptococcus mitis, Streptococcus mutans, aeruginosus, and sanguinis. So the rest of the upper respiratory tract is colonized by Moraxella catarrhalis, Neisseria species, and some diphtheroids. Now, others such as Haemophilus influenzae, Streptococcus pneumoniae, Neisseria meningitidis, and sometimes Staph aureus, they are all potential opportunistic pathogens that may also be found in the nasopharynx of healthy individuals. So ito po yung mga nabanggit ko kanina na opportunistic pathogens causing opportunistic infections. So they may be considered as normal flora or pwede rin po silang ma-isolate sa respiratory tract of healthy individuals. Pero ayun nga po, if the condition changes and it favors them, chances are they could still cause infections. So ayan po. Ayan, ito yung dito, yung mga nakalista po dito, yun yung mga usual na organisms na ma-isolate natin sa respiratory tract. Tapos yung mga next na nakalista po dito, ito yung mga ma-isolate natin minsan and they may cause opportunistic infections. Next po, the gastrointestinal tract. This is equipped with numerous defenses that are effective with their antimicrobial factors. Okay? So because intestinal pathogens are usually acquired by ingestion of organisms that are contained in contaminated foods or drinks, the host defenses throughout the intestinal tract, ayan po, there are host defenses throughout the intestinal tract. Okay, so despite the presence of these antimicrobial factors, the intestinal tract is colonized by about 35,000 bacterial species. But then again, lahat naman po ng yan ay commensals and may also be beneficial in the prevention of the overgrowth of potential pathogens. So pagdating po natin sa chapter 19 or on our discussion of Enterobacteriaceae, ayan po, madidiscuss natin yung mga pinaka-medically significant na members ng Enterobacteriaceae. Tapos, i-differentiate natin kung sino sa kanila yung mostly commensals and will only cause opportunistic infections and kung sino sa kanila yung talagang true pathogens. Okay, so that's on chapter 19. Next po, The kidneys, the bladder, cervix, and fallopian tubes are normally sterile, although a few organisms originating from the perineum can be found in the distal urethra, particularly in women. So, this is a fact that is more of an anatomical thing. So, women are more likely to suffer from urinary tract infections just because the anus is anatomically very close to the urethra. Ito po, nabanggit na natin to kanina. Lactobacillus is the most commonly known member of the normal flora of the genitourinary tract of women, and ang main reason po dito as to how they were able to establish themselves as an organism displaying mutualism is that lactobacilli can metabolize glycogen from the vaginal epithelial cells to maintain a low pH, which creates an environment that is inhibitory to many organisms. So para po sa mga lactobacilli, yung optimal growth pH nila na acidic can be harmful for other organisms. Okay? Ayan, next po is the microbial factors that contribute to the pathogenesis and virulence of bacteria. Okay, so pathogenicity, this is the ability of a microbe to produce disease in an individual. So an organism may be described as a true pathogen or as an opportunistic pathogen. So ma-idea na po tayo kung ano to kanina kasi kanina pa po natin to nababanggit. An organism that is described as a true pathogen. Ayan. True pathogens can cause disease in healthy and immunocompetent individuals at a high percentage of the time. Ulitin natin yun. True pathogens can cause disease in healthy and immunocompetent individuals at a high percentage of the time. So, whether kayo ay healthy, wala kayong sakit, hindi kayo immunocompromised, if the organism is a true pathogen, most likely po, they can cause a disease or they can manifest symptoms dun sa individual na may infect nila. Okay, so examples po ng mga true pathogens are Yersinia pestis and Bacillus anthracis. So Yersinia pestis and Bacillus anthracis, they are pathogenic in all situations, okay, or in nearly all situations. So when these species are recovered in clinical samples, their clinical significance is well established. Hindi tulad ng Haemophilus influenzae. So ang Haemophilus influenzae po, tulad na banggit natin kanina, they may or may not be pathogenic depending on the situation. Kasi po minsan, normal flora lang din sila nung respiratory tract ng pasyente. Okay, next are opportunistic pathogens. As I've mentioned earlier, organisms such as Staphylococcus epidermidis, under usual conditions, do not cause disease, but they can induce an infectious process in patients with prosthetic implants, like prosthetic heart valves. So minsan po, na-attract yung bacteria dun sa mga prosthetic implants ng pasyente. So mag-overgrow or i-colonize nilang area na yun, causing some sort of clinical manifestation. Ayan, for Haemophilus influenzae, it colonizes the upper respiratory tract of healthy individuals without causing disease. But if given the opportunity, this can lead to a life-threatening infection. Okay, terminologies naman po tayo. When we say nosocomial infections, these are infections that are acquired within the hospital approximately 48 hours after admission. Okay? So regardless po kung may nagawa na procedure o walang nagawa na procedure dun sa pasyente, basta may na-acquire siyang infection by just being in the hospital, that's already considered as a nosocomial infection. Kapag sinabi naman po na iatrogenic infection, these are infections that have resulted from any medical procedures or treatment that has been performed on the patient. So regardless po on the number of hours, it doesn't matter kung gaano katagal na yung pasyente dun sa ospital, basta po may infection na nag-develop dun sa body site kung saan ginawa yung medical procedure, that's already an iatrogenic infection. Okay? So, sana naiintindihan po yung difference between a nosocomial and a iatrogenic infection. Again, pag sinabing nosocomial, this is a hospital-acquired infection that occurs at least 48 hours upon admission, regardless or not na may nagawang procedure dun sa pasyente. Dito naman po pag sinabing iatrogenic infection, this is an infection that results from medical treatments or procedures, regardless kung gaano katagal na yung pasyente na naka-admit sa hospital. Next po, virulence. This is the relative ability of a microorganism to cause disease, or in other terms, this is referred to as degree of pathogenicity. So this is usually measured by the number of organisms necessary to cause infection in the host. So generally, organisms that can establish infection with a relatively low infective dose are considered more virulent than organisms that require high numbers for infection. Okay, so example po nito is these Shigella species. So Shigella species, they could cause disease with a very low infective dose. Approximately 100 bacterial cells lang po. Okay na, that's already enough to start an infection. Okay, so with that being said, Shigella is considered to be a highly virulent organism in comparison to other bacteria, which would probably need billions of bacterial cells before they would be able to start an infection. And next po is the ability to resist phagocytosis. So phagocytes, or phagocytic cells, such as macrophages and polymorphonuclear cells, they play a major role in defending the host from microbial invasion. So mamaya po sa last segment nitong discussion natin, we'll go through the 4 basic steps of phagocytosis. So tapusin lang po muna natin to. Okay, so there are many ways by which microbial species evade or avoid phagocytosis. And the most common mechanism for evading phagocytosis is that some microorganisms are capable of producing polysaccharide capsules. So meron po silang capsule on their surface. Na-discuss na po natin to last meeting, di ba? Capsules are polymers of polysaccharides that are firmly attached to the cell surface of the bacterial cell. Okay, so tinutulungan niya po yung bacteria to avoid phagocytosis by masking or covering the cell surface structures that are supposed to be recognized by the phagocytic cell. Kasi po, may structures muna na kailangang ma-recognize yung phagocytes natin before they actually proceed to phagocytize the invading organism. So kumbaga po, yung capsule, dinidisguise niya yung antigens or yung surface or the antigens on the surface of the cell para hindi siya ma-recognize and hindi mag-attach yung phagocyte sa kanya. Okay? Recall time. Recall muna po tayo on something that we discussed last meeting. What do we call the other surface polymer that is loosely attached to the cell and also helps with bacterial adherence? Okay po, sige, you may type your answers sa chat box natin. What do we call this other surface structure that is loosely attached to the bacterial cell wall that also helps with adherence? May natatandaan pa po ba from last meeting? Wala? Nobody remembers? Ayon, there's one correct answer. It's the slime layer. So, okay, yung slime layer po, both yung capsule and yung slime layer, this can help the bacteria to avoid phagocytosis. Pero yung slime layer po, it also gives the bacteria an ability to adhere to surfaces, making them difficult to get rid of. Okay, another bacterial structure that protects the organism from phagocytosis is protein A. So protein A, this can be found on the cell wall of Staphylococcus aureus. So this helps the organism avoid phagocytosis by interfering with the binding of the host antibodies to the surface of the organism. Okay? So to simplify this, kung ang dapat po na mangyari is yung antibody magbabind sa antigen that is present on the surface of the cell, dahil may protein A yung bacteria, makikibind din yung protein A dun sa kabilang side ng antibody para po hindi matuloy yung phagocytosis. Okay? Next, some organisms can evade phagocytosis by killing the phagocytic cell or by releasing potent materials in the tissues that can kill the phagocytes that are approaching. Okay, so pathogenic species of Staphylococci can release leucocidins that could cause lysosomal discharge of white blood cells into the cytoplasm. Okay po, so a staphylococcal leucocidin called as the Panton-Valentine leucocidin, this is lethal to leukocytes and contributes to the invasiveness of the organism. So pagdating po natin sa next week na po yung discussion natin on Staphylococci species or Staphylococcus species, mababanggit po ulit natin isa-isa yung mga virulence factors ng Staphylococci, tapos po malalaman natin kung anong ginagawa nila exactly or kung anong clinical manifestations yung magiging resulta na pinoproduce nila dun sa patients during the course of infection. Okay, ayan. Most infectious agents must attach to the host cells before infection would occur. So in virtually all other cases, the bacterium, virus, or fungi would require to adhere to the host cells before infection and disease can progress. Okay? So bago sila makapag-cause ng disease, kailangan makapag-attach muna sila dun sa ideal cell na kailangan nilang i-infect. Okay? So, the microbial surface structures that mediate attachment are called adhesins. So, kahit anong uri ng structure pa po yan, basta tumutulong sila in the attachment of the bacterium to the host cell, ang tawag doon ay adhesins. So, the main adhesins in bacteria are their fimbriae, pili, and also surface polysaccharides, which, as I've mentioned earlier, those are our capsules and slime layers. Okay po? So, isa sa mga nabanggit natin last meeting, no, is yung conjugation pili, which could be used for the transfer of genetic material. Yung mga bacteria po, meron din kasi silang pili that are not used for transfer of genetic material. Meron din silang mga pili na tumutulong lang for adhesion. Kaya po nabanggit din sila dito. Okay? So, fimbriae, these enable the bacteria to adhere to the host cell surface, which would increase the organism's colonizing ability and providing resistance to phagocytosis. Okay? So, may ambag din po sila in resisting phagocytosis. So, once the bacteria are attached to the non-phagocytic cells, phagocytosis by white blood cells is less likely to occur. Kasi po wala namang intention yung mga phagocytes natin to engulf our own cells. Okay, so an example of this is Escherichia coli. Some strains of Escherichia coli that can cause traveler's diarrhea use their fimbriae to adhere to cells of the small intestine. So pagdating nila sa small intestine, they would secrete a toxin that causes the disease symptoms. Another example is Neisseria gonorrhoeae. So Neisseria gonorrhoeae, its fimbriae is very important for it to infect the epithelial cells of the genitourinary tract. And isa pa po sa mga dahilan kung bakit hindi sila basta-basta nawawash away ng urine flow is because they are securely attached to the patient's cells thanks to their fimbriae. Okay, now some pathogens are able to survive within the phagocytic cell after they have been engulfed. So yung ibang mga organisms, iniiwasan nila yung phagocytosis or nag-a-attach sila sa mga cells para, ayun din, maiwasan nila yung phagocytosis. Pero may iba pong mga organisms na hahayaan lang nilang ma-phagocytize sila kasi po kaya naman nila na mag-survive within the cell. So some pathogens are able to survive within the phagocytic cell after they have been engulfed. So these organisms have developed different methods to prevent being killed intracellularly. Okay, so mamaya po, dun sa process of phagocytosis, explain din natin kung saang part or kung saang step ng phagocytosis yung killing and kung anong process ng phagocytic cells para ma-induce yung killing of the organism. Pero yun nga po, some bacteria are able to avoid that. Okay, an example of this is that some pathogens, ayan, prevent the fusion of phagosomes and lysosomes. Others have a resistance to the effects of lysosomal contents. So yung mga enzymes po that are supposedly released by the phagocytic cell to kill the bacteria, may resistance po yung mga intracellular organisms na to. And others po, they have the capability to escape the phagosome into the cytoplasm. Okay, so to establish itself and cause a disease, a pathogen must be able to replicate after attachment to the host cells. Okay, so okay, sige, nag-survive nga sila after phagocytosis, but now that they have entered the inside of the cell, the next step is for them to be able to replicate. So hindi po sapat na na-survive lang nila yung phagocytosis. Ang next step po dito is kailangan kaya pa rin nilang mag-replicate kahit na nasa loob sila ng isang phagocyte. So several pathogens such as Haemophilus influenzae, Neisseria gonorrhoeae, Neisseria meningitidis, they can produce immunoglobulin A protease or IgA protease that can degrade IgA that is found on mucosal surfaces. So di ba po, kapag sinabi nating protease, it can act upon proteins, and our antibodies, our immunoglobulins, they are proteins. So ayan po, yung mga nakalista po na organisms dito, H. influenzae, Neisseria gonorrhoeae, and meningitidis, they can produce an IgA protease. Other pathogens like Borrelia species, they can circumvent host antibodies by shifting key surface antigens. Okay, so the host, they can produce antibodies against the old antigens that are found on the surface of the cell, which are no longer effective because the organism has now new antigens that do not bind to the antibodies that were made against the old antigens. Naintindihan po ba? Okay, so during the primary or the first exposure of the pathogen, our immune system creates antibodies against those antigens that it was able to recognize nung unang encounter niya with the pathogen. Ang special capability naman po ng mga influenza viruses and Borrelia species is kaya nilang i-adjust or kaya nilang palitan yung mga surface antigens nila that were first encountered by the immune system. So kumbaga, kaya nilang magbihis or kaya nilang mag-disguise. So the next time na i-invade nila yung same host, hindi na effective yung mga naunang antibodies na nagawa ng immune system natin kasi iba na yung mga surface antigens nila during the succeeding exposure. Okay, next example po. Ayan, in most situations, when an organism is engulfed by macrophages, the lysosomal contents are released into the phagocytic vacuoles and then the organism would be killed. Kung hindi niyo po naintindihan yung sinabi ko, i-explain ulit natin yan mamaya dun sa specific step-by-step discussion natin on phagocytosis. But anyway, let me just repeat that. In situations where the organism has been engulfed by the macrophages, there is what we call a lysosome, and these are released into the phagocytic vacuoles, which contain the organism. Now, supposedly that should kill the organism. However, if the engulfed organism is not exposed to intracellular killing and digestive processes, then it is able to survive and multiply inside of the macrophage. So yung mga given examples po dito such as chlamydia, mycobacterium, brucella, and listeria species, ayan, they are easily engulfed by macrophages and neutrophils. Pero po, these species not only are able to survive inside of the macrophages, but they are now protected from the host's immune defenses, and they are also able to multiply intracellularly. Okay? So yung mga nabanggit na organisms po, aside from the fact that they were able to survive from the killing activities inside of the phagocytic cell, nagamit pa po nila to na opportunity para magtago sa loob ng phagocytes and away from the rest of our immune system. Okay, so this photo on the left shows us mycobacterium cells that are found within the patient's monocytes. Okay. Next, most bacterial exotoxins are composed of two subunits. So, andito na po tayo sa part kung saan may mga bacteria po na kaya nilang mag-produce ng toxins or enzymes, and this contributes to their virulence factors or their overall pathogenicity. Now, most bacterial exotoxins are composed of two subunits. So, yung mga exotoxins po, they are composed of kind of like two parts. So, one part is non-toxic, and it is only there to help bind the actual toxin to the host cells. And yung next part po, or yung next portion ng mga exotoxins, yun yung actual toxic part. So this causes tissue and cell damage, commonly encoded by phages or plasmids. Okay, so ulitin natin yun. The gene responsible for encoding toxin production is commonly encoded by bacteriophages and plasmids, which we have already discussed last meeting. Okay po? Okay, so exotoxins may be produced by both gram-positive and gram-negative bacteria, and examples of those have been listed here. Okay, so maging gram-positive or negative man po ang isang organism, if they have the gene to produce exotoxins, they will produce exotoxins. I-compare po natin yan sa production of endotoxins. Ang endotoxins naman po, they are composed of the lipopolysaccharide portion of the outer membrane of the cell wall of gram-negative bacteria. So, bali mga gram-negative organisms lang po ang may endotoxins or ang may kakayahan to produce endotoxins because it's literally a component of their lipopolysaccharide. Yung cell wall structure po nitong mga gram-negative organisms na to, yun po yung naging dahilan as to why they have an endotoxin. And this is not something that they can just produce. So the lipopolysaccharide contains three regions. So an O or an antigenic O specific polysaccharide, and then they also have a core polysaccharide and an inner lipid A. So kalimutan nyo na po yung dalawang naunang portions. Ang tandaan po natin dito na mahalaga is lipid A. So because of this portion, or the lipid A po, this is the portion of the lipopolysaccharide outer membrane that is responsible for the toxic activity of the endotoxin. Nagets po ba? Again, parang ang gulo ng explanation. Endotoxins, ulitin ko, endotoxins have three regions. The antigenic O specific, the core polysaccharide, and the lipid A. Hindi po mahalaga yung dalawa. Ang pinaka nagdadala po ng toxic activity ng mga endotoxins is the lipid A. Alright, let's now quickly go through the different routes of transmission. Medyo familiar na po siguro kayo dito kasi po sa parasitology classes nyo, meron din naman pong iba-ibang routes of transmission, iba't ibang mga parasites. Okay? So first up, this is applicable not only for bacteria but also a lot of viruses are airborne. Okay, so the respiratory spread of infectious disease is common, and this is generally a very efficient way to enter a host. So, di ba po, tignan niyo yung SARS-CoV-2. Diba? Very efficient yung transmission, and we don't really have to do anything to acquire the disease. We basically just have to go out without a mask at a crowded place and breathe for us to be able to acquire the infection. Which is why, ayan po, nabanggit natin na um, airborne transmission of pathogenic organisms is generally very efficient. Okay? So very small particles, or very small particles referred to as droplet nuclei, are the residue from evaporation of fluid from larger droplets and are light enough to remain airborne for long periods. Kasi po, often respiratory secretions are aerosolized by coughing, sneezing, and talking. Pero meron pa po tayong tinatawag na droplet nuclei. So again, ulitin ko yung definition. Droplet nuclei, these are residue from the evaporation of a fluid from larger droplets that are light enough to remain airborne for long periods. So this may possibly be one of the reasons kung bakit po laganap pa rin ang tuberculosis sa Pilipinas. Okay? So way before COVID-19 came and hit us, matagal na po nating problema ang tuberculosis. And this may be because of it being able to spread through droplet nuclei. Okay? So, scenario lang, no? Ano po yung pinakakadiring bagay na ginagawa ng mga tao sa labas? Diba? Hindi nakakaiba sa atin yung makakakita tayo ng mga taong dumudura sa labas, sa kalye, kung saan-saan. Okay, so now, those large drops of phlegm that has been coughed up by the infected person, they don't dry up instantly, di ba po? So what happens is that the fluid from these large particle droplets evaporate as time goes by. And the evaporation of those particles, or those tiny particles that are evaporating, they are now light enough to be carried by the wind and be airborne for much longer. So kung yung random person po na yun na dumura sa kalye, meron siyang tuberculosis, tapos ikaw wala, tapos na-inhale mo yung droplet nuclei na nanggaling dun sa plema na dinura nung random person na yun, this could possibly pass on the infection to you. So ayun po, yun yung konsepto of spreading infection through droplet nuclei. Ayan. So here are given examples. As I've mentioned, we have Mycobacterium tuberculosis. Okay. Next po, food and water. Transmission of gastrointestinal infections is usually a result of ingestion of contaminated food or water. So in some situations, infection occurs via the fecal oral route. Okay, so gastrointestinal infections result from the organisms that survive the harsh conditions of the stomach and competition with the microbial biota. And then they produce damage to the tissues of the gastrointestinal tract. Ulitin natin yun. Most pathogens that are able to cause gastrointestinal infections, kaya nilang mag-survive sa harsh conditions ng stomach, like the hydrochloric acid present in our stomach. Kaya rin po nilang mag-survive in competition with all the other microbial flora or normal flora na meron yung digestive tract natin. And there is a lot of them. Tapos, kaya rin po nilang mag-produce ng damage to the tissues of the gastrointestinal tract. So, doon po natin ma-observe yung mga symptoms such as abdominal pain, bloody stool. Ayan po. Okay, so organisms that can cause disease by means of pre-formed toxins that are produced outside of the body include Clostridium botulinum, Bacillus cereus, and Staph aureus. Okay po? So, they cause an infection with toxins na dala-dala na nila. They have already produced this even before they have infected anybody. Now, other bacteria produce a toxin only after infection of the intestinal tract. So initially, wala silang toxin na dala-dala, pero after they have successfully invaded the host's gastrointestinal tract, dun sila mag-start mag-produce ng toxins. Okay, so a commonly seen cause of diarrhea and intestinal infection is Escherichia coli. So this organism is a member of the intestinal normal flora. However, some strains of Escherichia coli can produce cytotoxins that can cause alterations in the biochemical activity of the intestinal epithelial cells. So yun po yung magre-result into problems with fluid and electrolyte control by the intestinal cells. Okay? So again, this is something na madidiscuss pa po natin in more detail pagdating natin sa chapter 9. And next mode of transmission is close contact. So all of the routes of transmission of infectious diseases require close contact, whether it be a respiratory pathogen or a virus that can be passed through kissing and exchange of saliva, or skin-to-skin transfer of diseases such as syphilis or impetigo. And obviously, ayan, counted din po as close contact ang acquisition of sexually transmitted diseases such as those that are listed here. So this photo on the left shows us a sample containing Treponema pallidum. Ayan, Treponema pallidum that has been stained with the Fontana-Mason stain. Treponema pallidum is the causative agent of syphilis, which is one of the sexually transmitted diseases that has been listed here. Okay, next, the classic example of bite wound infection is rabies, pero sa virology pa po natin madidiscuss yun. For bacterial pathogens, however, we have Pasteurella multocida. So Pasteurella multocida, this may be acquired from the bite of cats and dogs. We also have Echinella corridens, which could cause human bite infections. Okay? So kasama din po kasi sa normal flora ng oral cavity yung Echinella corridens, although not as commonly isolated, which is why it is able to cause human bite infections. Now, the photo to the left is a gram stain of Pasteurella multocida, showing its characteristic bipolar staining pattern. Bipolar. So if you would notice, ayan, mas dark po yung both ends nung cell as compared to the rest of the cell. So yun po yung tinatawag natin na bipolar staining or the safety pin appearance. Next, for the transmission with arthropod vectors. So arthropod vectors, ayan, some infectious agents rely on the help of arthropods for them to be able to transmit themselves among hosts. So infections that follow a mosquito, tick, flea, or mite bite is a common occurrence in many parts of the world. So listed here are examples of bacterial diseases that may be transmitted through the bite of their respective vectors. Okay? So as we go through the semester, iisa-isahin po natin yung mga yan, and we'll find out the exact species of their vectors. And the photo on the left shows us the Borrelia species under dark field microscopy. Ayan. The route of transmission, known as zoonosis, depends on the contact with animals or animal products. So certain organisms causing disease in animals may also infect humans who have contact with them. Ulitin natin yun. May mga organisms po kasi that can cause disease in animals. Pero po, if they are a zoonotic type of pathogen, kaya din po nilang matransmit to humans and cause disease in humans. Okay? So certain organisms that can cause disease in animals may also infect humans if they have contact with them. Now, these diseases may be passed on by animal bites like pasteurellosis na banggit na po natin kanina. Some of them could be passed on through arthropod vectors such as the bubonic plague. Some of them could be passed on due to contact with secretions like Brucella species, and others may be passed on through contact with animal carcasses and products such as tularemia and listeriosis. Okay, so this photo on the left is a culture of Brucella species on brain-heart infusion agar. Okay, so on to the host-resistant factors. Ang focus lang po natin dito will be on phagocytosis. So phagocytosis is an essential component of the resistance of the host to infectious agents. And this is the primary mechanism in host defense against extracellular bacteria and even some numerous viruses and fungi. So the cells in the human body that are responsible for phagocytosis, nabanggit ko na po kanina, are neutrophils, macrophages, and monocytes. Okay, so 4 activities must occur for phagocytosis to take place and to be effective as a host defense. So 1 is migration of the phagocyte to the area of infection, which is known as chemotaxis. Number 2 is the attachment of the particle to the phagocyte. Number 3 is ingestion, and number 4 is the actual killing. So unahin po natin yung chemotaxis, okay? Definition of terms, diapedesis, this is the movement of neutrophils between endothelial cells and also blood vessels. So when an infection occurs, massive numbers of neutrophils accumulate at that site of infection. So this accumulation is not a random event or this is not coincidental. Rather, it is directed, or this is a directed migration of neutrophils into the area where they are needed. So this migratory process is called chemotaxis. Chemotaxis happens because of chemotactic agents or chemoattractants, which attract phagocytic cells. So these include a number of bacterial products, products from damaged tissue cells, and even products from other responding immune cells. Now, your next question might be, Ma'am, ano pong pinagkaiba ng diapedesis sa chemotaxis? Are they the same? They sound the same because they're both a form of movement of neutrophils. Okay, so i-define po natin yung difference nila. Kapag sinabi pong diapedesis, this is the mere action of a neutrophil squeezing itself in between cells para makadaan siya. Okay? So it's not diapedesis if the cell doesn't have to force its way in between cells. Kapag chemotaxis naman po, regardless kung saan man dumaan yung neutrophil, whether it be in between cells or just in an open body cavity, where it doesn't have to squeeze itself through, basta po nag-move sila papunta sa area na yun because of a chemotactic agent, that is what we call chemotaxis because they're moving because of chemicals that have attracted them regardless kung saan man sila dadaan. Okay? So, naintindihan po. Next is attachment. Okay, so sa attachment po, the neutrophil membrane has various receptors and gawin po nating example yung immunoglobulin G or the IgG. So, ang mga neutrophils po, they have receptors for 2 different types of IgG, and these receptors can bind to the invading microorganism, resulting in the microorganisms being coated. Okay? So, ang mangyayari po, yung mga IgG ng pasyente, kakapit dun sa surface ng bacteria. Tapos, pag dumating na po yung phagocyte dun sa area kung nasaan yung bacteria, it can now attach to the bacterial cell. Dahil nga po, meron silang mga receptors para dun sa IgG na dumikit doon sa bacterial cell. Okay? So the coating of the surface of the bacterium with antibodies, as I've just explained, is known as opsonization. Okay? Next po, step 3 ng phagocytosis. The next step is phagocytosis or next step in phagocytosis is ingestion. Halo-halo na yung terms. Okay, so this process occurs rapidly after attachment. So the cell membrane of the phagocytic cell invaginates and surrounds the attached particle. So the particle is then taken into the cytoplasm and enclosed within a vacuole called the phagosome. So dun po sa area kung saan mismo nag-attach yung bacterium, magkakaroon ng space dun sa phagocyte that goes deeper and deeper until it swallows the entire bacterial cell. So magsasara din po itong space na to. And we will now call that as the phagosome. So the phagosome is the enclosed vacuole that has formed, which has the particle it just ate inside of it. Okay, so the phagosome will now fuse with the lysosomes, and lysosomes are vacuoles that contain enzymes and other antibacterial components. Okay po, so the combined structure is now referred to as the phagolysosome. So phagosome plus lysosome equals phagolysosome. Okay, so the list of enzymes that are found within lysosomes are quite long. So ang isang lysosome po, marami siyang laman. So it could include proteases, lipases, RNAs, DNAs, peroxidase, acid phosphatase. So most of these are important in the killing and digestion of the engulfed bacterial cell. Okay, on to the actual killing process. So the phagocytosis of a particle would trigger a significant increase in the metabolic activity of the neutrophil or macrophage. So this increase is termed as metabolic burst or respiratory burst. Okay, so the phagocyte will increase glycolysis, which would eventually produce lactic acid and hydrogen peroxide. So the hydrogen peroxide that has been produced at this time would diffuse from the cytoplasm into the phagolysosome. Ulitin natin yun. During phagocytosis, tataas po yung activity ng phagocytic cell in glycolysis. Sa glycolysis po, kasama sa mga products nito yung hydrogen peroxide. So ang mangyayari po, di ba meron na tayong phagolysosome? So yung mga na-produce na hydrogen peroxide due to the glycolysis of the phagocytic cell, magdi-diffuse po siya papasok dun sa phagolysosome. And this would help in killing the bacterial cell that has been phagocytized. Okay, so kaya po siya tinawag na respiratory burst is because glycolysis is an aerobic process. Okay, ulitin natin yun. Glycolysis is an aerobic process, hindi anaerobic. Okay? So glycolysis is space aerobic. Sometimes I feel like I talk so fast that I have to take the time to chop up my words because people might misunderstand. Anyway, so yun po, the killing process includes having a respiratory burst, which increases glycolysis, and glycolysis is an aerobic activity. Okay? Recall time. Recall po ulit tayo. Kakadiscuss lang din natin to last week. Ano po yung isa pang form of respiration maliban sa glycolysis? Okay, you may type your answer sa chat box. Which is, or what do we call the other form of glycolysis that can also be performed by aerobic and facultative anaerobes? And does anybody remember? 5, 4, 3, 2, 1, and okay. So, ayun po. It's called the Krebs cycle. Di ba po? Meron dalawang methods of respiration. So, glycolysis and the Krebs cycle, also known as the tricarboxylic acid cycle. So, as we have discussed last meeting, kaya po itong i-perform ng mga bacteria that are obligate aerobes and facultative anaerobes because these two processes require oxygen. Okay, and po, so here are the references that were used in the making of this discussion. Thank you so much po for listening. Our chat box is now open for questions. So does anybody have any questions po? Gising pa po ba kayo?